Hello and welcome everyone. In this podcast we're going to focus on deletions first and then duplications. There are some similarities between these as far as the effects they cause. Often we'll see that when we form a deletion, we often form a duplication. Usually when something is deleted, it usually ends up somewhere, and that's how we get a duplication often. So let's draw a chromosome. As we're talking about these chromosomal mutations, this will be a first time in a long time that we're not all that concerned with 5' and 3'. That might be a nice relief, right? So a deletion is as it sounds. You may have a chromosome here with these genes on it. A given chromosome is going to have many more than just 8 genes, but this is just an example. And this is its homologous chromosome, so they're going to have the same genes. They may be different alleles, but they're the same genes. Then what happens with a deletion is you can have one of two kinds of deletions. You can have a terminal deletion or an interstitial deletion. The terminal deletion is as the name implies or sounds. It's when the end of one of the chromosomes is deleted. End of chromosome is where the deletion occurs. So what happens with the interstitial deletion, instead of removing the end, we might remove D, E, F here, this region. So let's remove that. And now we're going to move G and H next to the C here. It's a new deletion that occurred in the middle of the chromosome. Now I say middle, of course that doesn't mean it has to be right in the middle. Just think of it anywhere but the ends. These deletions could include the centromere or not the centromere. If it includes the centromere, that's usually bad news, because that means that chromosome won't be segregated during mitosis or meiosis. Alright, the next question I want to pursue here is what happens during meiosis, right? Because you can imagine that we might have a bit of a problem here, and that is, how is this going to synapse during prophase 1 of meiosis 1? How is this going to pair up? Because remember, homologous chromosomes align and pair up or synapse with each other because of the similarities. But now we have this chromosome that's missing this large chunk. Is that going to be a problem during meiosis? Alright, so let's draw what happens here. Okay, so let's again draw our chromosome. I'm going to draw the deleted one first, AB, and then C, G, H. In this example, we've deleted genes D, E, and F. They're gone. Now the only way that we can make this homologous chromosome line up with it, remember it's A, B, C, D, E, F, G, H. Still has all of them. So to synapse, we're going to form a loop. And before we form that loop, let's remind ourselves that this being prophase, that we have two sister chromatids here. And same with here. What's going to happen is this D, E, F, I'm going to draw it, and it's going to loop out down here, and then G, H will shift over. So let me erase this part of the chromosome and show you what happens. So in this way, genes D, E, F, remember they have nowhere to bind to or synapse with, so they're looping out so that G and H can still align. Now I'm going to be drawing a lot of these kinds of chromosomes, and because of space confines and also so everything's still legible, just like your book does, I'm not going to draw the names of the genes on the second sister chromatid, but you need to remember that if it's A, B, C, G, H on this first one, it's also A, B, C, G, H here. Because remember, during S phase, it's copying it and it's making an exact copy. Same thing with this one. It is also this sister chromatid A, B, C, D, E, F, G, H. Even if I didn't write it, we know that because they're sister chromatids. Okay, this loop, now, if you looked at it under a microscope, a high-powered microscope, you could see this loop. And this turns out not to cause any problems at all. We can still have crossing over events in this area between the homologous regions. And so meiosis will proceed just as we've seen it proceed before. Now I want to pursue this next question, and it's an interesting question, and it's the question of how do deletions form? And so let's draw our sister chromatids, our homologous pairs of chromosomes with the sister chromatids, I should say. And in this case, I'm only going to draw 4 genes, A, B, C, and D. This is the one of the chromosomes. And now let's draw the homologous chromosome below it. Also with A, B, C, and D. They're homologous pairs, sister chromatids here in blue ones, and the sister chromatids are in red as well. During prophase 1, there's going to be crossing over between these two homologous chromosomes. One thing we didn't talk too much about when we were talking about meiosis is the fact that at the end of meiosis, we form gametes, sperm or eggs. And so each of these gametes will contain one of these sister chromatids. Let's write that down here. And there's going to be 4. At the end of meiosis, we always have 4. So you're going to have one, this blue one here. You'll have A, B, C, and D. And then a gamete that is formed in the second one will contain the other sister chromatid. A, B, C, and D. Then this third gamete that's formed will have A, B, C, and D. And then this last gamete that's formed will have A, B, C, and D from this sister chromatid. It's important to remember this as we move forward here. Now, also important to note here is if we did have a crossing over event, say right here, between these two homologous chromosomes, this top sister chromatid will still make this one, and this bottom one would still make this one. The two in the middle do an exchange. They swap these pieces of DNA. So it'd go A, B, C, and it'd come down here and pick D up. Technically, what we have here, it'd still be A, B, C, and still D, but it has this other part of the chromosome. And likewise here, we go A, B, C, it comes up, picks up the blue D. So that's going to look something like this now. In the end, it's still A, B, C, D, so no changes have been made. We'll talk later on how if we had a mutation on one of these, that could change it a little bit. But for right now, we're not going to talk about mutations. We're just talking about regular old genes. Now, this is how they're supposed to align. But if you remember, we talked about these things in our DNA called repetitive sequences. And they appear randomly in our genomes. We'll often find them between genes. We see them in the telomeres and we see them at the centromere, so they're found throughout our genomes. They're identical in sequence, so it's easy to imagine you could, as I coincidentally drew here, a crossing over event that occurred there. Now remember, I said this before, that the reasons that these synapse this way during prophase 1 is because there's a degree of sequence between the two homologous chromosomes. That's why they're homologs. Now because these repetitive sequences, let's write repetitive sequence here. Sometimes what happens during prophase 1, they don't align just like they're supposed to. It's possible that this repetitive sequence here could line up with here, with the second one, because they share sequence homology. They're the same sequence. So they could line up that way. Let's see what happens when we do that. Okay, now I'm going to redraw this chromosome, but this time I'm going to draw it so that there's a misalignment. And I'll show you what I mean here. We're going to have our genes A and B here, and we'll have our genes C and D here, just like we had before. And we're going to have our repetitive sequences here and here. Now remember, This should have aligned where A and B aligns with these two, and this repetitive sequence aligns here, and C and D with C and D on the blue, and this repetitive sequence here. But it's been shifted a little. It's not behaving very well. So let's say we have a crossing over event right here at this repetitive sequence, because remember they can align because all these repetitive sequences in this example are the same, so they match each other. The chromatids don't know they're making a mistake, but they are. Okay, now let's see what happens in our gametes. This top chromatid is at the end of meiosis 2 will end up in a gamete still. So it's still going to be A, B, C, D, just like we had here. So no change to that one. Let's look at the bottom one. A, B, C, D, no change as well, because both the top sister chromatid of this chromosome and the bottom sister chromatid of this chromosome were not involved in the crossing over event. But these two middle ones are. So let's follow the crossover and see what we end up in these two gametes. The mystery thickens here. So we pick up A from the blue, B from the blue, C from the blue, and then we cross down and pick up this part of the chromosome of the red one. Remember, it's physically exchanging the DNA. And then it picks up C and D. So blue ABC, red CD. So let's write that in here. A, B, C from the blue one, and then it's going to pick up C and D from the red one. This is called a duplication. We're going to talk about those next. Well, let's write it down here, duplication. Because we've just duplicated gene C. Now, let's follow the other sister chromatid we haven't talked about yet. And so we're going to pick up the red AB, and then we're going to follow it up here, and we're going to pick up the D. So we're going to end up with red AB, and then the blue D. We've just deleted C. So we call this a deletion. Whenever we make a deletion in this way, it's called, we always also make a duplication as a byproduct of it. We call this method of making a deletion, just write it across here, non-allelic recombination. We haven't used the word recombination too much, but really when we say recombination, we're just talking about crossing over. It's just another name for it. You can also form these deletions by having chromosomal breaks, which we're not going to talk too much about, but all that means is that you might have a situation where the chromosome breaks right here, and you end up with one of these terminal deletions. That's all we have for this part of deletions. Let's go ahead and erase this and move on to the next point. And let's talk about the different types of deletions. We can have a centromeric one. And remember, we said the centromeric deletions typically are rather problematic, because if they've lost the centromere on one of those chromosomes, it's going to be lost during mitosis or meiosis. The next one is a homozygous deletion, and we mentioned this one just a moment ago. And these are almost always lethal. Remember that these deletions, even though that's not the way we've drawn them, just because we're limited with how much room we have, but these deletions are usually large scale. Several genes are involved in deleting these, and it's often it's likely that if you delete both copies of a large region of the chromosome, it's going to contain genes that are essential, and if you lose them, the cells die. Now this other one are the heterozygous. Deletions. And these have varying effects. At first approximation, it doesn't necessarily seem like there should be a problem. If you have a chromosome like this, where this part has been deleted, say, let's say it's gene A, B, C, D, and E, and you have A, B, C, and you've lost D and E, you might think, well, we still have D and E on one of the strands, so that shouldn't create a problem. And sometimes it doesn't create a problem. but sometimes it does. And I'm going to talk about 3 instances where this causes a problem. And I'm going to need more room, so I'm going to erase this and start with a new board. So, heterozygous deletions. Why do they matter? Why are they problematic? Okay, the first one we're going to call pseudo-dominance. And I'm going to draw our chromosomes again. And let's say we have A, B, C, D, and E. And down here we have A, B, C, and that's it. D and E have been deleted. In most cases, if D and E are still present on the other one, there will be no problem. However, what if this person was a heterozygote for D? And the one that was deleted, if we draw it right here, right? Before it was deleted, it was D. And let's say this one was D minus. It's the mutant form. In that situation, it was okay because you still had this D. But now, once you've deleted this, you no longer have the unmutated D to back it up. You only have the mutant one. So this mutant one now behaves as a dominant allele. So we call it a pseudo-dominant. It shouldn't usually be dominant, but when you remove the unmutated one, now you're going to see the effect of this mutant gene D. Alright, the next one here, we're going to label as imbalance of gene product. And we can use our same chromosome example up here, except let's get rid of the minus by the D. Let's say that this is all the genes on this strand here are wild type, unmutated. Let's create a scenario here where there's this protein complex, and this protein complex contains 4 subunits. This is a protein, and let's call this protein X, and over here is protein X. And in order for protein X to have its job and be successful, it needs to bind with protein D. And it can do that because X is produced at a certain ratio, and having both copies of D, it's also produced at a similar ratio. And so you have this equal ratio of them, so as long as you have an equal ratio of them, you're going to more likely be able to bind 2 copies of each. However, if you've cut this in half, you're going to have less D proteins. And so you're not always going to be able to form this large protein complex of 2 Ds and 2 X's. You might be stuck with something like this, where you have your 2 X's, and since there's not enough D around, you might only get 1 D. And this creates a non-functional protein. There's an imbalance of gene product, in this case an imbalance of gene D. Okay, I'm going to erase these 2 examples here so we can talk about the third. And this third example is called haploinsufficient. This is kind of a difficult concept to learn, I think. A good example is looking at this Drosophila mutant called Notch. And then the wild type fly, just wild type here. And I'll do my best to draw a fly, but you know it's going to be horrible, so don't give me too much grief. Okay, that's my fly, but I'm going to draw the wing here. The wing is nice and rounded. The Notch fly, which almost looks like I guess a winged bear or something here, but it's supposed to be a fly. You're probably pretty jealous of my drawing. It forms a wing that is notched at the end. It's not supposed to be dramatically bigger, just the way I drew it so I could get the notches in there. So this mutation in the gene Notch causes this. And this mutation turns out to be a deletion, where on one chromosome you have the Notch gene here, but the Notch gene over here has been mutated and you no longer have it. So how does this work? And how is this an example of haploinsufficient? Turns out that Notch codes for a receptor. And what are receptors? Well, let's tell you what a receptor is. A receptor is something on the outside of the cell that allows communication between the external environment, or outside of the cell, and the inside of the cell. Various signals can get through. And when they get through, they go to the nucleus and they activate gene transcription. Certain genes get turned on. In the wild-type fly, just for convenience, I don't know how many receptors there really are, but let's say there are 4. All 4 of these are receiving that signal to help develop this wing. But if you only have 1 copy, maybe you only have 2 receptors. And so the signal is still able to go through, this signal from able to go through both receptors and able to activate gene transcription to make a wing. However, we have half the number of receptors now. We don't have a sufficient amount of receptors. And so because of that, you still make the wing, but it's not complete because we're not able to turn this signaling pathway on, this gene regulation pathway on in a sufficient manner. Still makes the wing, just not sufficiently. So this is an example of haploinsufficient. Okay, that ends the discussion of deletions. And let's move on to duplications. Let's go ahead and delete the word deletion. Now we're going to talk about duplications. Okay, let's draw our chromosome here. And let's say we normally have genes A, B, C, D, E, and F. As it gets duplicated through meiosis, like we showed before, we end up producing an altered chromosome that has genes A, B, C, D, and then C, D, and then E, F. So this region here has been duplicated. Again, why does this cause a problem? We're just making a few extra gene products, and by and large, duplications don't seem to cause as much of a problem as deletions. But they still can, and how they cause those problems is not quite as well understood. Though, one way has been discovered, and that is looking at this model organism called a zebrafish. And in this example, let's say that in order for the zebrafish to develop, it has this 3-protein complex. And let's say it takes protein A from gene A, protein F from gene F down here, and protein D. And as long as it has these 3 proteins, development occurs without a problem. However, as in this case here, if you have an extra D coming along here, so you have another D coming along here, binding to this complex, it almost serves as kind of like a poison. Instead of getting healthy development, we have development problems. They don't develop properly. So this is just an example of how, again, this protein imbalance problem, similarly as we saw with deletions, when you have an extra protein or subunit that binds to a larger subunit, sometimes having an extra one in this case can cause a problem. With deletions, having a less of a protein causes a problem. This is the opposite problem. More of that subunit, more of a problem. Now I want to talk about meiosis and crossing over. And that is, can synapses occur during prophase 1? Because remember, they have to line up properly. And we're going to see a similar solution as we saw with the deletions. Again, let's say we have A, B, C, D, E, F here. And let's draw our duplicated chromosome, where we have A, B, C, D, and then C, D again, and E, F, like so. Now remember, E and F want to pair up with each other. C and D do, but this extra CD here is causing some problems. And so the way this is solved, similarly as we saw with deletions, except now we're going to loop out this part of the chromosome. And in doing so, E and F can now bind with each other during synapsis, and this C and D can here, but the C and D that have been duplicated now loop out. Again, just as we saw with the deletions, this doesn't cause a problem. This is the solution to a problem. These will still get packaged up into chromosomes, into gametes. A, B, C, D, E, and F will go into one gamete, and this other one, A, B, C, D, C, D, E, F, will go into another gamete. As long as having this duplication doesn't cause any problems, there won't be any problems with that gamete. The last thing I want to talk about for duplications, and the last thing for this podcast as well, is to talk about evolution. So duplications and evolution. And let's define some terms here. A homolog would be genes with similar sequence. This is more of a generic term. We can break this down into more specific terms. Paralog and ortholog. Ortho means that these genes that are of similar sequence are a different species. So we'll put homolog genes in different species. If you get confused on how to tell these two apart, I always tell them apart because O here reminds me of other. So these homologs are in other species. Paralog. These are homolog genes, in the same species. What we end up usually calling all these paralogs, often fall into what we call a gene family. So let's talk about how we're going to make a paralog. So this is a recipe for how paralogs are made. And let's start with this assumption that we have gene A here. Gene A has an important function in this cell. We can even say it's essential. To the point that if you generated any mutations on gene A, that organism is not going to survive. So what could happen here then is we could duplicate gene A. So now we have two copies of gene A. And what this sort of sets up is kind of like this experimental gene that's happening in living organisms. Because remember, it's essential, so we can't mess with this gene. One of these has to stay unmutated. So this one is going to remain unmutated because it keeps performing that essential function. Now gene A over here isn't needed. It could be completely wiped away again and the organism's going to be fine. But over many generations, what happens is that we can start to take mutations on this gene. To the point where, at some point, gene A will become a new gene. So after many generations of taking these mutational hits to this gene, many generations, perhaps thousands of years, right? Gene A is still hanging on, still doing its job. But now gene A, its extra copy, for lack of a better term, we're going to call it gene A prime. It's still going to be quite similar to A, because it's not completely a new sequence. It's going to just have enough sequence changed that it can perhaps serve many of the same functions that A did, but maybe a little better, or maybe in a different way, in a way that's different enough to give this organism some sort of competitive advantage, some sort of selective advantage over others in the population. This selective advantage now will allow this new gene to persist in that population and to be passed on to the next generation. So now with that selective advantage, that can help direct evolution. This idea of how new genes evolve, because that's what we're talking about here, a gene that is evolving, is known as Ono's hypothesis. Alright, that ends this podcast, so let's close with a summary. So in this podcast we talked about deletions first. In our discussions of deletions we talked about how it works with meiosis and pairing up, how they form those loops. We talked about how these deletions are formed. We talked about different kinds of deletions, homozygous, heterozygous deletions. And we talked about how these different kinds of deletions can lead to different phenotypes, how some of the types of deletions almost always lead to a lethal phenotype, and how others lead to different kinds. Remember, we talked about pseudodominance, imbalance of proteins, and haploinsufficiency. Those are all good things to remember here. Then we talked about duplications. We mentioned that duplications are typically not as severe, but they can be, but typically they're not. We talked about what's going on with meiosis as well, as far as forming the inversion loops and all that good stuff. We talked about how they're formed, but really when we talked about how they're formed, we really did that when we talked about deletions, because when you form a deletion, you also form a duplication in the scenario we described. And we talked about some phenotypes. Again, with both deletions and duplications, intuitively, one might think that it shouldn't cause a problem, but we talked about how this could cause a problem with the protein imbalance. And then we closed with evolution, and that is how duplications allow new genes to evolve. That's in general what we talked about in this podcast for this summary. If you have any questions, make sure you come see me. If not, I'll see you in class. Bye.